at Convenant University, leading research on genetic variability and treatment of Africa men, helping to reduce disparities and promote precision medicine. Please, students, unmute yourself. Sorry, mute yourself. Genetic variability. A member of numerous professional societies, she is passionate about mentorship, STEM education, and female empowerment. Professor Olubanke continues to drive impactful research and global collaboration to address Africa's most urgent health challenges. Professor Olubanke, it is a privilege to have you with us today. Thank you for accepting our invitation and for joining this academic exchange. Ma'am, we are truly delighted to have you with us. Collaboration to welcome once again. And now, to formally welcome Professor Olubanke on the behalf of School of Pharmacy and Emerging Sciences, I would like to invite Professor Dr. Ravinish Mishra, sir, Dean of School of Pharmacy and Emerging Sciences, to say a few words. Over to you, sir.
Thank you, Madam Kritika. I welcome Professor Olubanke for accepting our invitation with respect to sharing of knowledge of her expertise to our students as well as faculty members of School of Pharmacy and Emerging Sciences. Welcome you, ma'am. Dr. Olubanke is known to me from last 13 years. I met her on eve of award ceremony that was held in San Antonio, Texas. The day was evening of 9th March that time, and Dr. Alu Banke is also one of the recipient of one of the most prestigious award from the IUTOX and SOT Endowment Fund, along with myself and remaining 8 more colleagues. From that time onwards, I used to see her contribution in field of research pertains to toxicology and environmental cancer and all these research areas through her professional association on LinkedIn. So I requested ma'am in last month, ma'am, if you could spare a time to share your knowledge, how we can collaborate, how can we do work together for the upliftment of the role of basic toxicology that is much needed in the country and in the world.
Sir, we can't hear you. I think there is a network issue. Ma'am, sorry for the inconvenience.
So, Madam Kritika, is it audible to you right now?
Yes, sir. Yes, sir.
So, also we have Professor Dr. T.R. Bhardwaj. Sir is known for his contribution in development of a new chemical entities from only university in India, that is Punjab University, Chandigarh. The molecule is known as chendonium iodide. Sir is having around 20 US patent. That is again a very remarkable achievement in field of pharmaceutical sciences. Apart from this, sir used to work on nitric oxide releasing hormones and paracetamol related molecules. Sir has achieved a lot in field of steroidal chemistry. Apart from this, polymer linking targeted drug delivery systems. So Dr. Bhardwaj has contributed a lot for the upliftment of this particular school of pharmacy and emerging sciences around a decade. So, Dr. Olubanke, in today's session, we have our faculty members as well as the students of our research, PG and UG project students. Apart from this, the limitation of this webinar is only 100 participants may attend this one. So we are also live on Facebook as well as on our YouTube channel. Over to Madam Kritika Bakshi. So I think she is not able to connect. So Dr. Alu Wanke, you can share your presentation and you can start with.
Thank you very much. I appreciate this privilege. I'm really blessed to be part of this great team. I'm trying to share my screen. Do let me know when it's very visible. I hope it's visible now.
Yes, ma'am, it's visible
Yes,
to
sir.
us.
Okay.
Yes,
So
ma'am, it
thank
is
you.
visible.
Thank you. Thank you very much. So good afternoon for people in India. I know it's after 2 now. Good morning from my students and other colleagues in Nigeria because we are still in the morning. And top of the day to everyone from other areas that might be in the evening and thereabouts. It's my pleasure to be part of this August gathering. I'm delighted to be invited to give this talk. I would like to acknowledge the presence of all the eminent professors, the Vice Chancellor of the Badi University of Emerging Sciences and Technology, India, the registrar, the eminent faculty there, my friend, the dean of the college. I want to appreciate everyone. I want to appreciate all the faculty, all the staff and students present on the call. I'll be talking on drug discovery in the 21st century, bridging the gap in natural products and synthetic chemistry, and where we are at the point. This is the 21st century. My name remains Professor Olubanke Ogunlana. I'm a professor of biochemistry at Covenant University, and other positions that I held had been read. So thank you. I want to say that the outline for my presentation is as described on the slide. We'll be looking at the global burden of disease and other historical perspective and where we are right now and some of the studies that I've done in line with the topic. Key definitions. Drug discovery is a systematic process of identifying biomolecules and developing them into therapeutics. So it's not just going to stop at the identification, it has to be developed. So it has several steps to the development. That's when it is translatable. However, the natural product chemistry is the exploration of biologically active compounds found in nature so that we can use them as a potential drug lead. And synthetic chemistry is the design of new or modification of existing molecules in the laboratory to create new compounds or to improve the properties of existing ones, such as potency, selectivity, solubility, and the rest. This slide, I just intentionally put it there because natural products is actually at the core of this presentation. So natural products are small molecules from biological sources with therapeutic potential. And there are examples, we have quite a number of them, natural products that have evolved into drug. We have example of artemisinin, we have example of pacitatel, which is the same thing as Taxol, We have quercetin from other natural products. So their strength is that they are highly diverse, and they have inherent biological activities that we have not even discovered, so many of them. However, their challenge is low yield, complex structure, and limited supply. And I have the structure figure one showing the structure of artemisinin and paclitatel. To start, I would like to talk about the global burden of disease. This is the motivation for new drug discovery. So I was able to find this particular figure two, that is tagged the composition of change in global and super regional life expectancy. So looking at life expectancy from 2022 to 2050, and we could see the life expectancy of people are increasing. Why is it increasing? Why are we having people live longer? Is because the gains of research in drug discovery is yielding. So we now have reduction in infectious diseases, reduction in maternal and neonatal disorders, reduction in respiratory infection. We are just coming out of COVID. We know how we are able to surmount and come out of COVID. So infectious diseases is gradually waning down. However, that is when, as people are aging, their metabolic disorders are showing up. So metabolic disorders such as diabetes, cardiovascular disorders, cancer is on the increase. So this particular chart, it's saying that there is high rising dominance of non-communicable diseases as people are getting older. And that's what it's saying. Heart disease, diabetes. However, we are benefiting from declining but persistent infectious diseases in low and medium income countries. So, and as a result, this is giving us high disease burden. So the high disease burden that was supposed to be coming down as a result of the victory we are having with infectious diseases is being negated by the high level of non-communicable diseases, metabolic diseases. And as a result, there is need to keep the tempo on. We can't afford to go down. There has to be more discoveries in order to meet up with the non-communicable diseases. And as a result, there is need for new therapies. So there's need for new therapies. The gain is have to be sustained, and for us to be able to improve on the gain and move beyond the projected life expectancy as determined in the global burden published paper here, we need to sustain and increase our output. And as a result, I want us to look at the evolution, the history of drug discovery. So we have the history from the ancient times when we talk about the Sumerian clay tablets that has the history of drug on the clay tablets, when we have the Pensao history of the foundation of traditional Chinese medicine, and when we have the Havoreda from the Indian, when we talk about the harmony between the mind, the body, and the herbs, and the papyrus from Egypt. So these are the ancient history, people that were able to document findings that they came about by trial and error, by intuition, by observation, by testing, and documented their findings. However, as that era wind over, we came to the era of classical findings, rational medicine. We were able to move from mouth to method to empirical findings. And this came about by the work of Homer, Hippocrates, Theophrastus, Celsus, and the Discorites. So these people are able to put up more rational discoveries into the manage of medicine, into healing. And they also documented their findings. And most of these findings were found within ancient Greeks and Romans. And as we move on from the era of rational, logical medicine, then we move on to the era of industrial revolution. And this era was coined from nature to laboratory. And that is the era of bioactive guided discoveries. So it spanned between 1950 to almost 2000. So this era spanned that long. And that was when we started extracting bioactive principles from natural products, testing for their efficacy in vitro and in vivo in animal, and then display presenting it into human. And the first one that I would like to talk about, even though I think morphine came before quinine, but the first one that resonates strongly with me is quinine. Quinine has been used before 1820 as herbs. Feel they have to keep planting the chicona barks several years. However, in 1820 it was purified. It was not the first alkaloids purified, but it was the first that has anti-malaria activity, and it was used to treat malaria in 1835. And after quinine, we have the history of accidental dye, aniline, by Henry Pelkin in 1856. This particular European was actually British, was actually trying to synthesize quinine when he accidentally synthesized this dye. It doesn't have therapeutic information, but because it was a story that resonates with quinine, then it's also equally important. So it launches the era of organic chemistry. And then going forward, we have the story of Felix Hoffmann that synthesized aspirin. So this was the first logical sentences. Okay, so when aspirin was when the salicylic acid was obtained from the willow bark, from the willow tree, he was the one that was trying to acetylate it to improve the biological activity, and then he was able to get aspirin. And this was in 1897. Thereafter, we have the activity of Gerhard Dogman in the 1930. He actually pioneered the development of sulfonamides by targeting the effect of dyes. And from there, he thought it was actually the dye that have the therapeutic activity. So his own dilemma was it has therapeutic activity in rats, but it doesn't have therapeutic activity in vivo, in vitro. However, because he was so convinced his daughter had a challenge, so he just loaded the daughter with a dye, and the daughter recovered. So it was later it was found that it was actually a prodrug. So it was when it is injected that it's metabolized into sulfanilamide that has the antibiotic activity. And this generated the era of sulfur drugs that we are still benefiting from till today. And then we have the Alexander Fleming discovery of penicillin that also was widely used during the World War II. And then this era is the era of empirical discovery. Lure us into systemic chemical synthesis and industrial evolution. However, after this era, we went into the molecular and genomic precision era. So this is called the target-based drug discovery. Okay, during this era, we have the discovery of DNA. So although discovery of DNA was not actually part of this era, but it gave a platform for this era. And Watson and Crick got a Nobel laureate award for their innovation, discovery of the double DNA double helix. And after this discovery, we also have the biotech revolution, the discovery of DNA technology in the 70s. And this brought about ability of scientists to manipulate and recombine the genetic material, and it brought about massive production of insulin, monoclonic antibodies, and other biological entities. After this era, then we have the completion of the Human Genome Project, which was completed in 2003. And this helped to have a blueprint of DNA blueprint of human, from where we know that we have approximately 22,000 genes, And out of these 22,000 genes, about 1,600 are disease associated genes. So this actually brought us into the target based drug discovery. And since then, after the rise of omics revolution, then where we have the I, that is integrated genomics, transcriptomics, proteomics, and metabolomics study, it provided an holistic evaluation into a system level understanding of disease. Unlike what we have before, we can target individual genes or we can target metabolite, but now ability to integrate all the omics gave us the system level understanding of the disease biology. And this created a translational shift. So we are no longer doing try and error, but rather we've been translated into the data-driven, predictive, and precision-based enterprise, linking genetic variation directly to therapeutic design. And we can be more assured of our outcome from our research. After this era, we are now in the computational and AI driven era, the era of predictive innovation. So it's not just about targeting and treating, it's about preventive, it's about predicting your risk and moving towards the prevention. And this is the era of integration of all of these things we've talked about, integration of biology, of chemistry, of data science, to guide intelligent data, the guided drug discovery. And also, we move into the artificial intelligence and machine learning models. So we have quite a number of these as open access and subscribe the version of it where we can accurate prediction of ligand binding and also admin properties. So this guide us to even know the risk before even getting into animals. So this, and because we now have the high throughput virtual screening, it enables large screening of compounds. What the previous time has done for us is that it has enabled us to have large reservoir of compounds. Some compounds don't even have meaning. They are just there and they are deposited in the virtual space. So with the high throughput virtual screening now, it enabled us to be able to do large virtual screening and test on targets that are just being known. And we don't need to go into the lab and spend so much money, but rather we can do everything virtually and then validate. Also, because of the omics technology that came up, integrative omics technology, we can also talk about structural biology and deep learning and leverage on tools such as the AlphaFold to revolutionize protein structure prediction and then be able to predict or understand drug target interaction. So all of this together now, and with the available platforms such as the Swiss admin lab, DeepChem, AlphaFold, we're able to use all of these platforms to enhance molecular optimization, target validation, and translational efficacy. So this era had helped us to have emergence of digital twin. So I can be one and I can have my twin online. I can have my twin in a digital space. And the manipulation that is done on my twin can actually help me to have a better, improved life. So this era had lead us to the emergence of digital twin, AI-assisted synthesis, omic-driven personalized therapies. As a result, ushering us into a new age of precision pharmacology. So what are the various potentials of AI in clinical trials? AI is transforming clinical trial landscape by increasing speed, precision, and inclusivity. So clinical trials is very key because after all of our discoveries, we still need to test in humans. So we have the clinical trial 1, clinical trial 2, clinical trial 3. It's after scaling all the three different protocol or operations that the drug can be registered and it can become translational, that is moving from the bench to the bedside. So how is AI also helping to transform that phase? It helps in these various perspectives: patient recruitment and trial design. So there are AI models that is actually helping to pick the right patients for the right trial. So it's not like before where everybody can just be enrolled into a trial and they will have failed trial. But now we can, because we are talking about targeted precision, system level. So the AI can help look at people that are best suited for a particular trial and recommend them based on their health record and recommend them for this trial. It can also help in real-time monitoring, safety monitoring. There are wearable devices now that can be linked directly to the online platform, and you can monitor real time the safety of the participants involved in clinical trials. It can also improve personalized and adaptive trials, and then automation of data management also is enhanced, and this also has helped to improve efficiency and enhance cost reduction. And the cost reduction can be as low as 30 to 50%. So we have examples of such tools. We have the Medita Data AI or Medidata platform. We have the DeepSeek AI. We have the InClinical. We have the Spot. We have the Pi Trial. So these tools are highly beneficial in clinical trial management or deployment. So what are the gains of modern drug discovery? It enhances data-driven, hypothesis-guided discovery. It reduces discovery time. So the normal time in the days of bioactive guided discovery, the normal time is between 10 to 15 years. However, with the 21st century discovery and innovation, the discovery time can actually be between 3 to 7 years. It promotes cost disruption, the reduction. The average cost for producing one drug is about 2 billion US dollars. And you can imagine a 50%, talk less of a 30% reduction in that. It promotes higher it to lead success rates, because you are not just starting in vitro where you spend so much money and then you have to test billions of ligands in the virtual space. No, you're actually in the virtual space. And because of the uh, complexity and the refining, the optimization of the virtual space, it's keep being refined on every time. So it helps to promote it to lead success rate. It promotes deep biological insights from omics and molecular modeling. So this time around now, even before the product get into the lab, we can predict the mechanism of action. It promotes personalized biomarker guided treatment. It supports predictive toxicology. It promotes integrative global databases, promote multidisciplinary collaboration. So this time around now, it's not somebody just sitting in the lab like the Fleming did and observed the growth of a fungus in a petri dish, in an agar petri dish. It's not going to be like that anymore. It's not one man thing. It's a collaboration, multidisciplinary, and it supports integrated natural, synthetic, and computational pipeline. So we're having integration of the different pipeline, and they are all very, very important. And all of these things we've been talking about had helped to fast track the delivery of our outputs from the natural products, which gives the natural scaffold, to translational research. So we say that from the bench side or from the field to the bedside. So we're able to deploy artificial intelligence, computational aided delivery, drug delivery into all of the various steps of drug discovery, development, and delivery to the point of care. And we are able to get value for money, value for time, and we're able to get the product delivered within a short time. What is in the picture post 2030? So post 2030, we're talking about intelligent and equitable drug discovery. So we are not just talking about 2025. We are talking about life to the 2050 and beyond. And what do we have? We have AI human synergy. There are fear that, oh, maybe AI is coming to disrupt the human environment, the ecosystem. No, the ecosystem is going to be enhanced. The artificial intelligence human synergy is going to bring in real-time design of adaptive, patient-specific therapeutic using generative AI and multi-omics integration. And then we're going to have a quantum and cloud computing, bringing out real-time clinical translation. So some of the challenges we are still having now is the computing power. By quantum and improvement in cloud computing is going to bring ultra fast stimulation and real-time translation. And then we have the era of gene editing and therapy, so that the treatment and precision treatment is beyond the protein. We're not just going to look at the protein, we're going to look at gene level, how gene can actually be edited through the CRISPR-Cas technology to give a gene level intervention. And then we'll be looking at digital health ecosystem, where we look at the integration of wearable biosensors, AI, therefore, AI diagnostics, and smart pharmacovigilance. And then this is also going to affect us, the global south inclusion, because we are talking about inclusivity, we are talking about precision, we are talking about genetic variation tribe or lineage linked. So there is need to bring everyone to the table. Everyone must be involved. So it's going to bring about inclusion. It's going to bring about open source data. It's going to bring about sharing of our equitable innovation. So this figure 7 is showing is a reflection of that. And this post 2030 is going to give us a shift from treatment to prediction and prevention medicine, because we'll be able to know our risk and we'll be able to quickly predict, so that we may not, even as our population age, the metabolic disorders, the non-communicable diseases that was supposed to be a factor for reducing the longevity will be prevented, because individuals are going to know their risk and prevent it. So we are going to be talking about preventive medicine, not only therapeutic. And I thought it's important to look at this. If we are saying that the drug discovery era had been improved over the years, and we're talking about the impact of genomics, omics, integration of the omics technology, and in the integration of the AI and computational method, how has this enhanced or the help to improve FDA approval? How has this helped to improve FDA approval? So I got this chart by Muller, published in 2025, early 2025, that talks about approval, the drug approval by FDA from 1994 to 2024. And from there, we can see that approval of a truth have increased. From 2010 to 2024, we can see approval of new molecular entity in the order of 459, approval of biological license application in the order of 164, all of them totaling 623. While before that, in 2000 and 2010, we've had in total 256. You can see the doubling effect as a result of the era of precision, the doubling effect as a result of AI and computational guided discovery. I want us to also take a look at some case studies. So some case studies, I termed it from nature blueprint to predictive drug design, the future of translational research. And the first one I picked, even though I told you that we can also have nicotine, but the first one I picked is quinine. Quinine was isolated from chicona bark as far back as 1820. But I can tell you that the synthesis of quinine did not come up until 1944. And that is when we can have the synthetic of its analogs in post 1950. Before then, they have to always plant a lot of chicona tree and extract quinine from the back of it. And the synthesis of quinine brought about the era of malaria control and inspire malaria pharmacology. So we could see figure 10 showing the structure of quinine and its derivatives. And then I also want to look at aspirin. Aspirin was derived from the willow bark with the willow tree, and it was synthesized, it was improved by Felix, working for Bayer Group then in 1897. And he was just acetylating, he was just trying to acetylate different things. And he saw that it improves the tolerance of salicylic acid when it was acetylated, and it was marketed as aspirin. So synthesis work together with natural product discovery. Without synthesis, we've seen that the limitation of natural product discovery is there. We can't keep extracting from the plants. It has to move beyond discovery to synthesis for us to really have the industrial revolution of it. And we can see the figure showing salicylic acid and acetyl salicylic acid. And I want to look at how has computational aided drug discovery help in improving aspirin. So aspirin is not a selective, it's not a uh, COX selective inhibitor. It affects both the COX-1 and cyclooxygenate 1 and cyclooxygenate 2. However, with the advent of computational aiding, virtual screening, aid-guided virtual screening, it has led to the development of other selective COX-2 selective inhibitors. We have them as the new generation selective COX-2 inhibitors. And these are here in the figure 2. We can see how that was done. They were able to differentiate between the COX-1 and COX-2 from the amino acid sequence. And from there, they were able to see that one has isoleucine that has the larger side chain, and the other has valine that has a shorter side chain. So all they just need to do is to look at bigger molecules that will not fit into COX-1, but will fit into COX-2. And this was done virtually. And this brought about new generation selective COX-2 inhibitor, improving the side effects, improving the therapeutic effect of COX inhibitors. And also with the advent of AI now, the drug repurposing is coming up. And then there are indications for how antimalarials, aspirin analogs can be used as anti-cancer, for cardiovascular issues, for neuroprotective use. And then I also want to talk about artemisinin. Artemisinin was extracted from Artemisia annua, the sweet wormwood. And it was from a project by the Chinese government and by this researcher that eventually got Nobel laureate for it, Tu Youyou, in 1917. And his mechanism of action is that he increases free radical generation inside the plasmodium infected red blood cells, as a result, destroying the plasmodium parasite that causes malaria. And this also had a lot of issues with respect to its synthesis. So it was not stable. So however, they were able to use semi-synthetic derivative to improve the stability and solubility of artemisinin. So if you look at the original artemisinin, you can see how science was able to add more variants to improve the stability and the solubility of the original artemisinin. Okay, that is the marriage between natural product discovery and the synthetic chemistry. However, AI too had been optimized to generate the next generation antimalarial. So we now have some the antimalarial that is currently being optimized. What is the advantage of this new AI generated antimalaria? It has longer half life. Artemisinin has very short half life, 7 days, but this one has longer half life, and then it's also reduced resistance. And these are generated with AI optimization. We also have the paclitaxel, is popularly called Taxol, and is also an intervention from the US NCI Natural Product Screening Program in the 1960s. And the uniqueness of this particular compound is that it's from the back of the Pacific yield tree. And this tree takes about 100 years to grow. So how do we generate this tree to have sufficient Taxol for cancer patients? And the quantity in the back is very small. So and it helps. It brings a lot of revolution to cancer treatment because it binds and stabilizes the microtubule. Microtubule is normally disintegrates under cell division, but when there is a molecule that binds and doesn't allow it to disintegrate, then it helps to prevent all cell division, and it also induces apoptosis in cancer. So it's a very potent anti-cancer agent. However, semisynthesis has to be developed for people to be able to use it because of the scarcity of the tree. Still talking about the marriage between the natural products and the synthetic chemistry. So the Taxol also has been improved by rational design from natural lead. And now we have the improved version from computational modeling. We have the Taxoterol. This is a synthetic analog that has emerged as clinically superior to the semi-synthetic analog. It has higher solubility, better efficacy in breast and prostate cancer. And we can see the computationally aided Taxoterol. So it's from the parent compound Taxol. So I want to just quickly use the next few minutes to talk about our contribution to the 21st century drug discovery. I want to say that when I started my research life at the university here at Covenant University, I started with working on natural products. I'm interested in getting biomolecules from natural products. And I started with bioactive guided natural product discovery. Because of the limitation of that research facility in my university and in the country, I got a fellowship. I went to China for a whole year to do bioactive guided discovery. I took plants from Nigeria. I went with over 1 kg extracted material, and I did bioactive guided discovery. And it led me to the isolation of over 50 compounds. However, because this knowledge has not diffused so much, then I have to spend months upon months in the lab testing for the efficacy of my plants, of my extracts and my compounds on cancer cell lines and antimalarial cell lines and the malaria plasmodium cell lines. I had to do that. And that, after several months in the lab, led me to the publication of these two papers to my left. However, when I came back and this era of computational discovery came up, I started going back, checking those compounds and subjecting them to computational aided discovery. And even much more compounds came out promising, and we are doing more work on them. So that is how we can tune ourselves, our research to the 21st century research and be able to get better value for our efforts, for our money, and for the global knowledge. So I have quite a number of my students' work. So I didn't allow what happened to me to happen to them. So because I work on quite a number of natural products. So what they do, once they hit into the lab, they first do systematic review, get the gap in literature, get into the lab, assay for the phytochemicals in the plant, and match it again, go into the in silico modeling, and you can see some of the papers that were published from that effort. And then we then also try to see targets that these compounds are suited for from the in silico modeling, and then we come back to the lab to test them because we are biochemists. We have to always validate our findings. So we come back to the lab to test their mechanism of action. And from there, we have been able to publish a lot of papers. We have also been able to see the mechanism of action of some of our compounds. So our research integrates in vitro, in silico, and in vivo molecular approach to predict and validate and optimize bioactive compounds from natural products for potential therapeutic application. I also want to talk about some of my research interests in the area of vector biology. This my student, also a PhD student that have graduated with me. We always start with systematic review or the traditional review. And then for this particular study, because we know that the target had to be correct, so we had to leverage on a machine learning algorithm that was done by somebody in bioinformatics that was part of the group. So the person validated the machine learning algorithm in Drosophila. So my student had to validate it in Anopheles gambiae because our interest is vector biology. And from there, she was able to identify potential targets. And she tested those potential targets by subjecting them to RNA interference, trying to use RNA interference to block them and monitor the survival of mosquito after the target had been blocked. And some of these papers are what are reflected on the slide there. And I still have about 3 or 4 PhD students that are currently working with me now, trying to validate some of the target that she had already identified from the work she did. So we are looking at the effect of each shock protein, elongation factor, trialase, and arginase as a potential insecticide target for control of malaria, for vector control. Also in the area of biomolecules discovery, we are also interested because I belong to a group called the Prostate Cancer Transatlantic Consortium. And that group, I'm the PI at my university. With that group, we are interested in knowing why the prostate cancer is highly endemic in our Black population men, and also how it is highly associated with high level of death, mortality. So we are looking at biomolecules from Blacks, and we are looking at potential targets, druggable drug targets that can be novel to us. And that has brought us to all of this. We did a machine learning approaches to genome-wide association study in Blacks, which was one of my students did and was published. The student in the US now, Enoma. And then some of my students also try to look at the association of some of these targets. And then this is to ensure that we are locally relevant because we can't wait on the global north to solve our problems. We have to start from the global south. And this is also a part of my research under synthesis. Funnily enough, I did masters in pharmaceutical chemistry. So for my master's project, I synthesize metal complex. So I complex zinc with quinine. I complex zinc also with chloroquine, and I monitored their ability in the lab, their enhanced activity in the lab. And I published that as far back as early 2000. However, I'm doing a similar work now, but I'm not going to go through the blind synthesis method anymore. What we did was we did in silico, we identified the targets as triskitolase. We did in silico modeling. We got our contact in the UK to express the gene, the protein for us, transcriptolase in human and transcriptolase in plasmodium. And we subjected our compounds. So I also have contact in one of my sister university here that Obafemi Awolowo University, a chemist that is into synthesis. So we are working together. So he synthesized the potential compounds from our doc, from our in silico heat. And then we tested this compound in the lab. And the next target is going to be optimized. So we are not going to stop there. So there will be optimization because this has not been published. There will be optimization. And from optimization now, we are going to come back to the lab to test. It is after all of this testing using the protein that we will then test in the blood infected in the cell line, and then we test in animal, and then we can move forward from there. So that is allowing the improvement in the drug discovery to affect research environment and process. So what are the shared challenges in translational drug discovery in low and medium income countries? The reason why I call it shared, because I know I'm presenting for an institution in India, and I think these challenges are shared to us. So we have limited and inconsistent funding for translational phases. It's not cheap. We talked about $2 billion. If it's even reduced, it's 1 billion. So we need money to do translational phases of research. We talked about inadequate integration between wet lab findings and computational validation, minimal adoption and knowledge of artificial intelligence and machine learning for modeling. We talk about weak infrastructure and limited access to high performance computing. We talk about weak intellectual property protection and patent translation mechanisms. We talked about fragmented collaboration between academia, industry, and clinical sector. So if these different units are fragmented, that means I may not be able to know what is prevailing in the academia. The industry may not know what I'm doing. And then the clinical sector will not trust my output in the academia to be able to test at the bedside. So all of these challenges lead to long regulatory timelines for approval of novel natural product-based drugs. So what are the opportunities to bridge this gap that have been identified? We've talked about the innovation that will come out as a result of the 21st century drug discovery. We've talked about where we are in the global south. We've talked about the challenges that is mitigating against our translational tendencies. So how do we bridge these challenges? There should be enhanced academia industry regulatory collaboration. We can't run away from it. So for the industry, I would say clinical space also. For me, maybe hospital, okay, because we're talking about health related. So this collaboration has to be enhanced. There should be government support for translational research. There should be harmonized regulatory pathways. There should be regional pilot trials. Within the global south, we can begin to pilot trials. We should not always lean on the support of the global north to be able to have our novel discoveries. We should train our people so that we can pilot trials and be able to come up with our own indigenous new product. We should encourage collaboration between India and Africa. India is leading in the global south. India is leading in technology, is leading in manufacturing. We have a lot of products from India in Nigeria. However, there is need for that collaboration to be able to enhance and improve inclusivity. In conclusion, The drug discovery in the 21st century relies on the integration of natural products and synthetic chemistry. Natural products is the anchor for creating the scaffold that synthetic chemistry can work on, and then that the targeted era and the precision era can rely on. So if there is failure at the natural product side, then there will be bias in whatever is coming out. There are still a lot of natural products that have not been tapped into, a lot. If we look at most of the discoveries have been intentional government direction, the artemisinin, your Chinese government looking at their books, the NCI for Taxol. So intentional, the scrutiny brings about novel discovery. Then there should be advances in AI, synthetic biology, and chemoinformatics. And these advances should be tapped into by the global south so that we are not left behind. These advances should be tapped into by the global south. Capacity building is a key. Since a lot of students are on this call, capacity building is key. I should not say because I'm a biochemist, maybe this 21st century discovery is not for me. No, I can learn, I can collaborate. Even at my age, I am always learning new things. I'm not old anyway, but you can imagine students that are 20, 25, less than 20. So that means the post 30 we are talking about is still there. It's still for all of us to tap into and take advantage to ensure our translational innovative products. Innovation must align with global and regional health needs to develop safe, effective, and affordable medicine. We should stop doing research that stops in our computer or on the shelf. We should conduct our research to translational innovation so that it can be able to deliver safe, effective, and affordable medicine and improve our world. I want to list this following points as my recommendation. The global South, the collaboration between the global South should be strengthened. India being a leading of the region to take the lead, and there should be strengthened collaboration. And we can start with the University of Badi University of Emerging Sciences and Technology, collaborating with Covenant University. We could intentionally start research space that could strengthen our collaboration and project us towards translational innovation. We should expand AI-driven prediction toxicology and virtual screening platforms. We should invest in infrastructure for high throughput and computational modeling. I want to actually praise India because as I was doing the research, I've been using IMRAT, Impact. But this is the first time I will know that Impact actually means Indian Medicinal Plant Archive or Hub. So it was because I was doing the research, I was trying to see what we have, what we don't we have. I said, yes, Indian is actually the leading of the global south. There is need for more of such infrastructure or computational space being projected and being made open access. There should be collaboration between academia and industry for drug development and commercialization. Most of the open access repository that are open, most of the repository that houses natural products, a lot of them are not open access. I want to praise Indian. Some of the ones in Africa are also open access, but some are not. So as much as possible, it should be open access to encourage collaboration, to encourage the spirit of sharing our innovation. And then there should be development of regional training programs on computational pharmacology and bioinformatics to train our young ones. You should not live in silos anymore. I'm a biochemist. I just have to understand things that buttress on cells, proteins. It's beyond that. It's beyond that. We should develop regional training programs to enable us to be able to maximize our potentials. We should promote inclusive regional funding and IP policies and knowledge that support global South innovation. And I have this following as my references. And I want to appreciate the God Almighty for this privilege. I want to appreciate the management and leadership of the Badi University of Emerging Sciences and Technology, India, for the kind invitation and opportunity to share my thoughts on the emerging topics, drug discovery in the 21st century. Special thanks to my friend, Professor Rabin Mishra. Thank you for watching out. I was so surprised when you were able to identify me after so many years of first meeting, the Dean of School of Pharmacy and Emerging Sciences. I appreciate your warm invitation, collaboration, and continued academic partnership. Deep appreciation to Covenant University and the center that I belong to, the Covenant Applied Informatics and Communication African Center of Excellence, is a World Bank funded center of excellence. This center of excellence had changed the dynamics of research in my university. It was funded by World Bank through the intervention of our government, and with about $7 million being plunged into the university for staff, for training of students, and for infrastructure, we're able to have so many opportunities, so many infrastructure to do research. I'm also grateful to our collaborators and partners, most especially the Prostate Cancer Transatlantic Consortium, for their ongoing collaboration in prostate cancer and molecular oncology research. My heartfelt thanks to my students, all of them, my research associates, and all our co-authors, our clinical scientists too, because they give us open space, into their lab, and we can interact with the patients. For their consistent dedication to integrating computational modeling and natural product pharmacology and toxicology into our research efforts. Great appreciation to all the international collaborators, my colleagues, mentors, participants for advancing our shared vision towards a sustainable, predictive, and inclusive global drug discovery. I want to say that together we can accelerate equitable data-driven drug discovery for the global South. I remain Ogunlana Olubanke from Covenant University. Thank you very much.
Thank you so much, ma'am, for your valuable knowledge and inspiration. Now is the time for question and answer session. Please go ahead and ask your questions to Professor Olubanke. Anyone, participants?
Hello. Good afternoon, ma'am. Myself, Deepika. I'm having a question. So, my question is how can AI and machine learning helping us nowadays in identifying novel natural compound with a therapeutic potential?
Thank you very much. There are so many sites online, and a lot of them are open access. Like I've said before, that the major repository for the indigenous innovation with respect to synthetic chemistry is natural products. So over the years, there have been a lot of repository storing medicinal plants, like the one I mentioned now, Impact by India, is one of the leading. So anytime I want to do in silico, I also go there. So if you go to Impact will be able to tell you the different compounds that have been extracted from different plants. So it's not just going to stop at the different plant. It can talk about the bark, the leaf, the twigs, the fruits. So you can then be more specific. So you can do that. Go to all of those sites. and it's not only Impact, we also have Dr. Duke. So you can go to those sites, download your compounds, get to PubChem, get their molecular name, maybe in Smile, and then you can do molecular docking. So molecular docking now can help you to understand, but you must have had a target. So if you're only like for me, sometimes I start with, if it's in cancer space, I start with getting the target. There's a work I'm currently doing now. Okay, but because I'm not a computational scientist, so I have my students in that space and other colleagues. So what we do is that we ask for data from the Biobank, UK Biobank. So UK Biobank over the years has stored data of their people, and there are genomics data, there are proteomics data, there are transcriptomics data, there are metabolomics data. So we can with their permission, they will give that data, and it's free of charge to the people within the global south. So somebody already paid for it. If not, if it's people from the north, global north, they will have to pay. So we can have that data, and then my colleagues in the computational space will work on the data, develop algorithm, work on the data, and then be able to understand the unique targets that are common in the blacks. So if our interest in prostate cancer, for instance, so we'll work with people that have been diagnosed with prostate cancer. So we'll look at the unique data that are the unique target that are common to them, and they will present those unique target to us. So because I do the work in the molecular space, I can take patients. I have archive of patient blood sample with me that I've extracted DNA and RNA. I can test those samples to see if the target that they have identified, maybe mutation in that target, is also common within the space in for people for prostate cancer patients in Nigeria, and compare with non-prostate cancer patients. And then if it's a druggable target and it has implication, like I do androgen related metabolism or response. So if it has implication in that particular pathway, then we can look for targets. So we can dock it with targets already identified in the maybe the Impact or Dr. Duke, that's ligand, and then we can dock it to our target and then start our work from there. After we have done all of those, and we can test for toxicity on adment lab or sweet bed, and then we can then come to the lab. So that way we will have reduced 1 billion, 1 million compounds to maybe 10. And then we can come to the lab and start looking at it. And then because I also have a hub where my compounds can be synthesized, I can share with them and they can synthesize for me, and then we can continue the work. That is the way to do the research in the 21st century now, not any other way. Thank you.
Thank you so much, ma'am.
Hello, ma'am. I have a question. I am Shalini.
Yeah.
Ma'am, is there any AI-driven technology to screen phytoconstituents from the plant?
Please, can you repeat that question?
Is there
Can you
any
repeat?
AI-driven technology to isolate or to find out the number of compounds or the phytoconstituents from a new entity or new plant material?
Oh, thank you very much. So AI can only build on what we have given it. Computer is garbage in, garbage out. It can only build on what we have given it. So that is why the work on natural products cannot afford to stop. There are still millions, billions of chemical entities that have not been discovered. So we cannot stop the discovering process. So the database is there, but the database uh, was able to mime all the work that has been done over the years to build the database. So it's whatever is in the virtual space that was used to build those database. So for us to get novel compounds, for us to get new compounds, the natural product is still the only scaffold that provides that knowledge. So we can't afford to stop the work on natural products. The synthetic chemistry can help tweak what is already existing based on the interaction within the in silico model. They can tweak, they can add, they can look at the pharmacophore and change and add OH and add acetyl, but they may not be able to completely come up with a new scaffold. So if you look at the drugs that have been generated over the years, they are all circulating within the alkaloid region for anti-malaria and the rest with N nitrogen, and for flavonoids for antioxidant molecule and the rest. So, but the best way to get novel compound is still through natural product engagement. Thank you. I don't know if I
Thank
answer
you,
your
ma'am.
question.
Thank you, ma'am.
Any question from anyone?
Yes, ma'am. Good afternoon, ma'am.
Good afternoon, good
I
morning.
have a question from you. Can synthetic chemistry improve the properties of natural drugs?
Can you repeat it?
Can synthetic chemistry improve the properties of natural drugs?
Very correct, yes. In fact, if you look at the history of natural products, synthetic chemistry had played a major role. A major role. So what do you want to talk about? Is it quinine? When quinine came, it has a lot of side effects. So a lot of analogs of quinine was synthesized. We have chloroquine, we have mefloquine, and even though some of it had been advised against malaria treatment, but they are still being used for other things. So we talk about Taxol. So we have other molecules that came up, and so synthetic chemistry help us to introduce other functional groups that help improve solubility, bioavailability, efficacy of natural product discovery. So they are very key. So most times, if it's also difficult to synthesize, like chloroquine took more than a decade, more than a century to be synthesized. Okay, so the synthetic chemistry also help to ensure that the product is synthesized, because we can't keep taking it from the back of Chicona back. We are going to run it into extinction. So synthetic chemistry are key. So synthetic chemistry, natural product scientists, computational scientists are to work together to ensure that the gains of 21st century towards the effective drug discovery is maximized. Thank you.
Thank you, ma'am.
Good afternoon, ma'am. I have a question for you. How can collaboration between chemists and biologists to speed up the drug discovery?
Okay, thank you very much. So I'm going to use my own example. I collaborate with so many people. I have chemists. I have the chemists that work with me in my university here, and I also work with some chemists in another sister university, Obafemi Awolowo University. So I work with computational biologists, bioinformatics. I even work with AI. I work with our group here. It's called they are in the engineering department, information communication engineering. So my friends there do work with federated genomics, cloud assessment, and AI. So I work with all of those people, so that if because I can't get all those knowledge on my own. What I have, nobody can take it from me. But I have to be relevant even in the 21st century drug discovery. I can't use the same model I use for my PhD anymore, where I had to extract over 50 compounds after spending hours in the lab and then have to spend another 1 year to start testing each of them through different cell lines. That cannot happen again. I have to leverage, I have to maximize, and I have to ensure that I'm able to save my efforts and increase my efficiency. So the only way I can do it is to collaborate. So whatever I have, as I'm working, I'm sharing with my colleagues in bioinformatics. I'm sharing with my colleagues in IC, information and communication engineering. I'm sharing with my colleagues in chemistry. So we do meeting together, we troubleshoot, and we enhance. So, like for instance, if I'm not able to use AlphaFold, they have the mastery to use it. So they will use it and give me feedback that I want. The synthetic chemist is able to give me my virtual ligand, is going to make it available for me to test in the lab. So there is no way in this century that any researcher can work alone. You work alone, you die alone. You work together, you maximize, and you are able to bring about translatable innovation. Thank you.
Thank you so much, ma'am.
You're welcome.
Anyone wants to ask another question? Anyone? Thank you so much, ma'am, for
Good
sharing
morning.
knowledge with us. May
Thank
I
you.
now warmly invite Professor Dr. T.R. Bhardwaj, sir, Advisor to Chancellor, to kindly share few words. Over to you, sir.
Thank you, Kritika. Good morning, Professor Olubanke.
Good morning, sir.
And first of all, I would like to congratulate you for your very informative, motivative deliberations pertaining to your talk. We at Badi University are located in the hub of pharmaceutical companies, and as you know, that India has acquired third position globally, but in the areas of formulations and bulk drugs, not for the new chemical entities, not for the drug discovery. We have very limited discoveries. Coming to your areas of specialization specifically, in which you are exploring, particularly the anti-malarial, anti-prostate cancer, or BPH maybe, and similarly anti-diabetic also. So these are very thrust areas which are being explored by you. And as we know that these days, the two-third drugs which are being used clinically, they have their origin from the natural products. They have been modified, they have been derived from natural sources. And we in India, we have one traditional system of medicine which is known as Ayurveda, in which herbal drugs are used. Although some of the components or the constituents of these drugs have been isolated and maybe to some extent explored also for their evaluations, but these days, on account of the facilities available, on account of the technology advancements, on account of the other like docking studies, there is again chance, opportunity to explore those molecules with respect to the docking study so that we can minimize or identify the lead and then can develop the area accordingly so that we can have minimum wastage of time and money also. It's very costly affair to develop a drug or to invent a drug. It takes thousands of rupees and maybe time also. As you said that out of 10,000 molecules, only 10 would reach to the clinical stage. And if out of 10, one could see the clinical applications, that's more than enough what we have. So we are specifically in your terminalia, what was the structure of the compound which was isolated or which was explored? Do we have that structure also? Am I audible?
Yes, we can hear you. We can hear you. Can you repeat the question again, sir?
Yeah, with respect to your, you see, antidiabetic drug which has been isolated from Terminalia plant, what were the components or the constituents of those structures or the
Yes, sir. So the work that had been done with Salmonella, we didn't do proper extraction. So what
I
we
see,
did was we did GCMS,
I see,
uh, so and LCMS. So
I
they are not novel compounds. They are
see.
compounds that are already available in the literature. So
Mhm.
because of the infrastructure deficiency that we have, so most of our students are not able to completely do the total isolation from medicinal plants. Oh,
I see.
okay, okay. That was what I did, but I had to travel all the way to China to be able to
Mhm.
do that. So, so the compound that was extracted for Terminella was actually compound that was already existing, but
Mhm.
all the same, we saw that it has very good anti-diabetic activity.
I see.
It has very good anti-diabetic activity, but that research has not been further explored. So I didn't bother to move towards synthesis. So we're just trying to show at that point that because a lot of our people in Nigeria, because of the high level of poverty, and because a lot does not really believe in the orthodox medicine, so
Mhm.
the a lot still rely heavily on medicinal plants. So sometimes we try to validate their claim and be able to say, oh, these are the possible actives in the plant,
I
why the drug is probably being used as an anti-diabetic. And these are also the mechanism of action of those actives. So for that, my student, that was what the student did, not extracting novel compounds.
I see. And secondly, we are also working in the area of prostate cancer, specifically benign prostate hyperplasia,
Okay.
in which we are preparing, synthesizing 5-alpha reductase inhibitors.
Oh.
5-alpha reductase are involved from the hydration or the hydrogenation, from testosterone to dihydrotestosterone, which is responsible for its proliferation of the prostate gland. And we have synthesized numerous compounds having structure pertaining to the finasteride type, which was clinically used maybe for 2-3 decades, and the new it's a new what called the analog dutasteroid which has come up. So that's again one of the area, and which is again prevailing in old age, specifically the prostate cancer or enlargement, which is again some drugs are required in that area also. You see. Of course, we have surgical approaches, but they are not being preferred by the patients also. Thirdly, as you said that we in India, we would also like to have collaboration with your university and specifically with you so that we can share our expertise, we can share our knowledge, we can work together to come to some conclusion and explore some areas where we can have new entities, new chemical entity, new drugs, because chances are there. Only I think fraction of natural products have been explored, and rest are again pending. They are not being explored, although they are having some therapeutic activities also, which have been reported in our old what you call the traditional books. So yours this thing collaborations. I would like to suggest and request Professor Ravinesh Mishra also to initiate that program in this direction, so that we can share our expertise and work together and come up with some positive results for the betterment of humanity, betterment of treatment, better drugs, safe drugs, free from toxic expectancy. Thank you very much once again. Thanks. Over
Thank
to
you very
Pooja.
much, sir. Thank you. I actually want to say that Indian is leading within the global south. Indian is leading. And when I look at the history, I was like, how come something has not come out like what Chinese did? from India.
Mm.
India is known to have very rich resources of natural products.
Yes.
If there is an intentional drive, I'm sure we will come out with novel chemical entities, new chemical entities. So if you look at the way that artemisinin came, it has to be intentional. Government asking them to look into the hive. The NCI for Taxol, it came out from intentional effort. If we have such intentional effort also in India, known to have very rich reservoir of chemical entities. I mean, the traditional medicinal plant documentation. I'm sure a lot of new chemical entities will come from India. And I think that's the spirit to go, Sam. And I'm saying that we're also happy to collaborate with you. I also work in the aspect of androgen receptor pathway. I work on that aspect. I'm new. I have not done so much research on it. But like I told you, the way we are looking at is to look at what will benefit the blacks. Because when whites are doing their precision targeted medicine, even in the clinical trial, even in the in the start of the work, they look at their people. So if you have blacks in the population, it's just be like 2%. So such a drug, if it's developed, may not be viable in our population. That is the more reason why it is important that governments should rise up, especially more of our African governments, so that things can be done right. We can prevail. We can protect our legacy, and also show our global relevance. Thank you very much. We'll be happy. I personally will be happy to collaborate with you. Thank you.
Thanks for your thankful statements. Over to Kritika.
Thank you so much, sir, for your kind words. Thank you so much, ma'am. As we approach the conclusion of today's webinar, I take this opportunity to express our sincere gratitude to Professor Olubanke for sharing her time, insights, and expertise with us. Ma'am, your presence has been truly enriching, and we deeply appreciate your valuable contribution. I also thank our advisor to Chancellor, Professor Dr. T.R. Bhardwaj, sir, Dean SPS Professor Dr. Ravanish Mishra sir, faculty members, organizing team, and technical support staff for making this event possible. A special thank you to all the attendees, students, scholars, and colleagues for your participation and engagement. With that, we officially conclude this session. We
Kritika,
hope.
Kritika, just one minute.
Yes, sir.
We would like to give some time to the Professor Ravnish also. He wants to have some clarifications or some collaborative research which we are thinking to, which has been again considered. So let Professor Ravnish Mishra speak something before you conclude it or before we can disperse.
Okay, sir. Okay, sir. Over to
Over
you,
to
sir.
Professor Mishra.
Thank you, Dr. Olubanke, for insightful lecture. And there is, you told our audience as well as the faculty members, there are a plethora of ways to do the research and how the natural product remedies can be beneficial to the mankind that already we have seen during this COVID-19 pandemic. Because this natural product or natural remedies act as a preventive measures during this COVID-19 pandemic. In India also there are a means combination of various herbal formulations are widely adopted by the most of the populations, and they got treatment and they got prevented themselves by using this natural remedies, which is the actually the part of our old tradition system, that is our Ayurvedic system. So here in India, the Ministry of Ayush and CCIUMB basically working on this natural product research uh, area. And you will be pleased to know that in our team at School of Pharmacy and Emerging Sciences, we have a very renowned scientist, Professor Dr. V.K. Kapoor. He has contributed in the means herbal compendium, means compiled by the government of India. So eh, worked a lot on this validation protocol of various herbal formulations. So this is our means first interactions, and we are exchanging our means what we can do together. So we will also have some discussion with our core team of this research wing, Dr. Bhardwaj, Dr. Tejvi, Dr. Kapoor, and our herbal scientist, Madam Salni, Madam Archana, and our head, Dr. Bhartendu Sharma, and few other colleagues. So we have a pool of scientists, those who used to work on this natural product research. So, ma'am, in next meeting we will come with some ideas. And already, ma'am, you have told about this the discovery of anti-cancer molecule paclitaxel, and how you can do it wonder by doing the some synthetic approach, that is the example of a docetaxels and other many more drugs. So, ma'am, here in India, we are means as told by Dr. Bhardwaj. So Dr. Bhardwaj is also having expertise in means hydrochloroquine, that is the kind of a drug of choice during the primary prevention of COVID-19 pandemic. And Bhatti University has contributed a lot to all these manufacturing setup in India, providing them the accommodation at that time. So, ma'am, you rightly said means your university convent and Bhatti University, at least let's start sharing our views and let the students as well as faculty get aware on the equal platforms. So already I had spoken to Dr. Bhardwaj also earlier to this lecture, so and told about yourself. Then Dr. Bhardwaj has given his consent. Definitely we are going to work with the Olubanke and going to have a lecture of series of her expertise and some professors from your schools. We can have a miss exchange pool. And ma'am, anytime if you feel like to visit India, you are most welcome, ma'am, at our place. We are happy to welcome you here. And also we can collaborate in writing book chapters, in writing research paper, because the way we do research here, that is very similar to your place only. So so thank you, ma'am, for all these words, and we have delivered a very means insightful lecture that cover each and every aspects of means the role of this natural product chemistry, ma'am. So thank you so much. I would also like to thanks the organizer behind the scene, Madam Kritika Bakshi, Madam Alka Sharma, Madam Priyanka, and our online contributor, Madam Akshita Kapila, Mr. Pansul Jamwal, who has been supported a lot for this conduct of this webinar. And in last, also I will thanks to our management, Honorable Secretary, Governing Body, Mr. Gauram Junjunwala sir. Our Honorable Vice Chancellor is a agro scientist, Professor Dr. Jatindra Kumar Sharma sir. Our worthy registrar, Dr. Kusmeet, and the person who has worked for this successful conduct of webinar. So thank you, ma'am. Means keeps on connecting. Are going to have such insightful lectures from your side. So thank you, ma'am. Thank you so much. Over to Madam Kritika.
Thank you.
Thank you so much, sir, for your kind words. Thank you so much once again, ma'am. Your presence has been truly enriching, and we deeply appreciate your valuable contribution. With that, we officially conclude this session. We hope to host many more such knowledge sharing events in the future. Thank you all once again and have a wonderful day ahead. Thank you to all.
Thank you, ma'am.